Hello everyone. So as I was talking about, I graduated from Valencia Community College in 2018 with a degree in biotechnology, my AA. Next slide. So my very first kind of introduction to biotechnology was actually in high school. I was a laboratory assistant for a Project Lead the Way kind of curriculum where we would teach students kind of some actual laboratory techniques, working with cell lines, working with plasmids, doing some pretty cool like experimentation, and I would help set up those experiments. And because of that work, I was awarded a scholarship from the program director that kind of helped me along in my initial college studies. Next slide. So by the Valencia College, where I was studying biotechnology, it was again a very hands-on class where every day we were in the lab. There was lecture, but it was also it kind of would apply to like whatever technique we were learning at the time. That then culminated into a poster presentation on a knockout plasmid that we changed from, if those of you who are familiar with GFP or green fluorescent protein, using PCR to change that into blue fluorescent protein. Very cool project and kind of introduced me to a number of techniques that would help me in my later career in analytics. During that time, I actually pursued what's called the BASE credential, the biotechnology accreditation credential that kind of solidified all of the techniques that I learned while I was at Valencia, and then really gave me a nice platform to kind of introduce myself to some other biotechnology laboratories when I decided to move to Gainesville, Florida from Orlando. That's when I kind of started getting involved with this mentorship and presentation. I find a lot of joy in sharing this story with other students. So after kind of earning that credential, in the next slide, I gained a internship to complete my degree at the University of Florida's Microbiology and Cell Science Department. This actually, despite what I think my initial impression of academia was actually a pretty rigorous environment. I was there every day, 8 a.m. to who knows whenever the project would be ready. But I was very happy for that exposure and because it actually culminated in my publication here. I have kind of a small snip of it. So I was working with an archaeon, which is a component of life that we don't often study, just characterizing a particular way that it respirates, allowing it to live in very high salt, high pH environments. I designed an assay to kind of evaluate whether or not organisms that had this particular respiration would be able to survive in high UV environments as well. So I became third author on this paper, and very, very prestigious and great opportunity to continue my studies in analytics specifically. It kind of helped me determine that I liked answering questions and designing experimentation to answer those specific questions. So because of the connections I made during that internship at UF, I decided I really liked Gainesville. And after some work in Orlando, and I kind of go into that in the next slide, while I was in Orlando, when I completed my degree, I still wanted some more leadership exposure and some more organizational techniques. So I worked for a reference laboratory called Aventis Biolabs, where I was the manager for an accessioning department. And what we did there was essentially intake patient samples from doctors' offices, hospitals, and some research laboratories, and just performed like a number of different testing panels that they would request. So that was urinalysis, blood panels, and some genetics sampling as well. I designed a database to help, because we were pretty much just using like paper copies and then giving paper to our departments on what kinds of testing they wanted. I designed a system to kind of intake those forms, make sure that they were accurate, make sure that there was like a validation process to ensure that we were in compliance with, I think that it was CAPA accreditation for that particular lab, and assisted with the like individuals who would visit to make sure that we had those processes in place. So I appreciated the leadership aspect, but there wasn't really a lot of actual like lab work associated with that position. So when an opening came for the genetics department that was expanding, just to be a scientist there and help develop the new instrumentation, the new methodologies, I kind of stepped down from that position after training someone up into that leadership role, and started working in the genetics department exclusively. There I had some exposure to what's called liquid handling systems, and that kind of would far away be a really great exposure for my later career. On the next slide, so the wet lab experience was really great at Aventis, and I wanted more. Because of my experience with my internship, I moved back to Gainesville, Florida, and joined Brammer Bio, which is now Thermo Fisher. It was later, I think a year after I joined, it was there was an acquisition by Thermo Fisher. So at Brammer, I was exclusively in analytics, kind of, as I mentioned, like continuing to answer questions for different departments in our laboratory. We were exclusively working with viral vectors and for gene therapy products. So clients from different labs would come to our lab and want to upscale their process and have our analytics department develop new assays to make them faster and more precise and accurate. So some of the assays or testing that I did for that was cell-based, so a lot of infectivity, some nucleic acid-based methods like PCR, and more specifically digital droplet or ddPCR. And along with kind of the wet lab work, I continued some of my leadership exposure by leading projects. So that would mean kind of coordinating the testing that and resulting that clients were looking for, presenting proposals on new assays or new instrumentation, and training new associates or technicians rather on those methods that we would develop and define in our group. So during my 2 years at Brammer Bio / Thermo Fisher, I was promoted to a scientist 1 position, and I was very grateful for that opportunity and more exposure to a number of different techniques within the lab. And I was very excited to kind of continue that development aspect, like looking for new assays, new cutting edge technologies to introduce to our laboratory. So after 2 years there, I decided to move on to the next slide. Oh, pardon. Yeah, so this is just a kind of a highlight on some of the techniques that I learned while I was at Brammer. So those can be a reference later. I can definitely answer questions during the open time. So if I could have the next slide. So the next in my current role after Thermo Fisher was a company at the time. Again, these biotech companies get plucked up pretty quickly. It was originally Ology Bio that was then acquired by National Resilience, and this is where I currently have my role as a research associate in the assay development team. What we do here is determine new technologies, new techniques, try to improve the techniques that are already here, or establish new methods for clients that are coming in. The big key difference from my last role to this one was within the biotechnology space. I was previously working exclusively with viruses or AAV, and here we work with a number of different platforms from monoclonal antibody production to different viruses for the purposes of vaccine and gene therapy products. So with that kind of comes like a whole plethora of different techniques and methodologies to learn to kind of suit each of those different platforms. So still within the same, trying to answer questions, trying to make things more efficient and precise, and training individuals on how to execute those particular testing methodologies. So my focus now, in addition to kind of bringing on those new technologies, is trying to employ new automated systems. So way back when I had that initial exposure to robotics and liquid handling, that really kind of carried me through a small red thread through my career towards automation and robotics. So at present, what I'm trying to do is employ a new robotic system called the G Station to kind of take some of the liquid handling or the pipetting out of our methodologies here, so that we can kind of keep scientists doing, the thoughtful design of their experimentation, as opposed to just like pipetting from liquid to liquid. So I still do have exposure and do have some wet lab work with some of these techniques, but my focus now is trying to evaluate new technology, new automation, and bring that to our lab here.